The journey of a new medicine inside the body is remarkably complex, marked by a crucial event: its meeting with a target, typically a receptor. These receptors are specialized protein molecules often found on the surface or inside cells. Imagine a city full of doors, each with a different shape. For a drug to affect the body, it must find the right door, fit into its unique lock, and either open it or block it. Consider the story of Sarah, a medical student who learns that drugs can act as keys. In her mind, she visualizes medication molecules searching the vast cellular landscape, looking for the right lock, a receptor with a specific shape that matches only certain substances. One day in the lab, Sarah observes a simple experiment using acetylcholine, a natural neurotransmitter. She sees how it fits perfectly into the muscarinic receptor, like a key slides into its lock, causing the cell to respond. The door swings open. When another molecule, atropine, is introduced, the response vanishes. Atropine, though similar in shape, enters the same lock but instead jams the mechanism, keeping the door firmly shut. These interactions can lead to either activation, agonism, or inhibition, antagonism, of the cell's response. Sarah ponders the implications. Sometimes, drugs fit so well that they trigger the full response, the classic agonist. Other times, drugs only partially activate the receptor, acting as partial agonists, opening the door halfway. Some, like atropine, serve as antagonists, blocking the receptor and preventing any activation at all. It fascinates Sarah that in the body, competing keys, natural molecules, drugs, and even toxins can fight over the same locks, their effects depending on their concentration and affinity for the receptor. She also encounters the concept of allosteric modulators. Instead of fitting into the main lock, these drugs attach at a nearby site, bending the lock just enough to make it easier or harder for the original key to work. This subtle influence can fine-tune the cell's response without directly opening or blocking the door. Receptors themselves can change. With prolonged exposure to certain drugs, they may decrease in number, downregulation, needing more drug to achieve the same effect. Or they may become hypersensitive, a process known as upregulation, where even a small amount of medication produces a marked response. By the end of her class, Sarah realizes that drug-receptor interactions underpin nearly every effective medicine can have. Therapies are crafted, and side effects appear, all because of how well a drug fits a receptor and what happens after the connection. Like city doors opening and closing in response to different keys, the microscopic communications between drugs and receptors shape health, disease, and healing in the human body.